and Jay, an assistant professor of medicine at the Rutgers Robert Wood Johnson Medical School. Kyle received his PhD from Virginia Commonwealth University and did postdoctoral training at the Wistar and University of Pennsylvania, and then at Moffitt. The title of Kyle's presentation is Protective Immunity in Ovarian Cancer is Governed by T-cell Intrinsic Mitochondrial Stress Responses. Kyle.
Yeah, thank you, Ned, for the introduction, and good afternoon, everybody. So the focus of my work really is to understand the immunobiology of ovarian cancer, with our approach being to facilitate the rescue of anti-tumor immunity to better protect these women as we implement new therapeutic approaches. So many of you probably know that ovarian cancer is essentially a lethal disease. There has been some improvement in the survival benefit of these women over the years, mostly due to the introduction of PARP inhibitors, but women with advanced epithelial ovarian cancer, hover around a 29 to 30% survival, 5-year survival rate. And it's important to note that the majority of women who are diagnosed at these late stages. So clearly, there are therapeutic needs that need to be met so we can better manage the treatment of these women. And I believe that immunotherapy holds a great promise for this. And in fact, it's changed the paradigm of management of cancer greatly, more greatly than since the advent of chemotherapy. So really, we're trying to interrogate how immunotherapy may be utilized for these women. And this really stems from work done by my postdoc mentor almost 20 years ago now, where his group, in association with George Kukos, were the first to show that ovarian cancer really was an immunogenic disease. Interestingly, it was shown at that point that women who had infiltration of T cells, and it's obvious to us now, but back then it wasn't so clear, that had infiltration of T cells into their tumor beds had a improved outcome. It was a quite dramatic improvement, actually. And interestingly, the spatial location of these T cells was critical for these outcomes. T cells that infiltrated intraepithelial tumor islets essentially protected these women, whereas those T cells that infiltrated stroma had no protective benefit. And over the years, we've learned that the T cells that infiltrate tumor islets actually possess the greatest antigen specificity against tumor antigens. So that's basically the rationale for thinking about how we can leverage immunotherapy for treatment modalities. And it's interesting to note, as a side note, that not only T cells are important. I know that we're a very T cell intrinsic field in cancer immunology at this point, but B cells have also been shown to be critical in regulating and protecting women as well. So that's also an active area of interest for cancer immunologists, specifically focused on ovarian cancer. So we know that ovarian cancer is immunogenic from these data and from data that I'm not discussing here. But the current class of immunotherapeutic agents has really had, disappointing results to further protect women who do have T cell infiltration into their tumor beds. As an example, checkpoint immunotherapy is, it's the hot topic now, and it's had quite a degree of success in the management of patients with lung cancer, for example. Or this is an example of a clinical trial from non-small cell lung cancer, actually. And blocking PD-1, which is a checkpoint molecule upregulated on T cells that when it's engaged with its ligand, PD-L1, is known to suppress T cells, essentially. So antibodies have been developed to block this interaction, and in lung cancer, utilizing Pembro, this has a clear survival benefit for these patients. However, in a similar trial in ovarian cancer, there is essentially no survival benefit. Actually, only around 10% of patients respond to this type of immunotherapy. And this is the same story for CTLA-4. It's a similar story utilizing cellular therapies as well. CAR T cell therapies and TCR engineered approaches have all generally led to poor responses in patients with ovarian cancer. So our central question is, how can we understand the immunobiology of ovarian cancer to improve these outcomes? So I have basically two objectives that I'm going to share with you all this afternoon. The first, I'm going to review some previous work that we've done very briefly, just understanding the role of a different class of checkpoint molecules. It's not necessarily related to metabolism, but I think it's critical to understand that the immunobiology of distinct tumors may be unique enough that we need to understand how checkpoints may be differentially expressed and how they may differentially regulate anti-tumor immunity. And the second point, which I'll spend the bulk of the talk because I think it more closely relates to our objective of understanding immune metabolism, is to, and this is really a work in progress. So this is happening in real time. So it may be a little granular, but hopefully it'll be enlightening and interesting for the audience. And that's the elucidation of this molecule that we're interested in. It has a really kind of horrible name, to be honest, UHR1BP1. So we're interrogating this molecule and how in trying to understand basically its biology and how it may regulate T cell activity in ovarian cancer specifically. So as I mentioned, I will give you a little bit of a background on some of the work that I've done in the past to understand immune regulation, specifically in ovarian cancer. A few years ago, we became interested in butyrophilins because they are they're on chromosome 6 within the major histocompatibility complex. They have a large degree of structural homology to other known immunosuppressive molecules such as PD-L1, and they are also within the B7 family of co-inhibitory and co-stimulatory molecules. There are a few different isoforms within the butyrophilin 3A family. We became specifically focused on butyrophilin 3A1 due to its, it has similar structural homology to PD-L1, which, as I mentioned, is a potent immunosuppressive molecule that's utilized by a variety of malignancies to escape immune surveillance. So we began to interrogate this molecule to understand how it may dictate and regulate immune responses in ovarian cancer. And as you can see, we did Western blotting and quantified expression of this molecule, butyrophilin 3A1, and observed that in patients with high-grade serous ovarian cancer is obviously dysregulated when we compare this to normal tissue as well as to benign tumors. Generally, in my thinking, anything that's dysregulated in cancer is, good for the cancer and bad for the host, but we didn't really know yet what the function of this molecule was in the context of this malignancy. As you can see, its expression is generally regulated to epithelial ovarian cancer cells. We've also found that it is expressed on leukocytes, with the majority of it being expressed on myeloid cells. So when we utilized immunohistochemistry to quantify expression of this molecule over 200 patients with high-grade serous ovarian cancer, we basically generate two cohorts which express differential levels of butyrophilin 3A1, and we had clinical annotation. So we were able to define that the expression of butyrophilin 3A1 was associated with outcomes for these patients, in such that those patients with less butyrophilin 3A1 expression within their tumor beds had a better overall outcome. So was this related to any kind of anti-tumor immune activity? That was a hypothesis that we were generating at the time, and we began to pursue that through some in vitro studies. I won't get into the details of how this was done, but essentially when we presented this molecule, butyrophilin 3A1, to T cells from healthy human donors, that it severely inhibited the ability of these T cells to proliferate when upon activation. as you can see in the red here versus the black, which is a large degree of proliferation through dilution of this proliferation dye. If we assessed the release of type 1 cytokines such as interferon gamma is a very prominent anti-cancer cytokine. So T cells that produce this are generally considered to have anti-tumor potential. Those that were exposed to butyrophilin 3A1 also had a decreased ability to produce interferon gamma. So this is quite an immunosuppressive molecule. So that was exciting for us. This is a highly expressed in cancer, and ovarian cancer is a potential therapeutic target. So we then generated antibodies using phage display. This is actually yeast. This is a yeast 2 hybrid system to create antibodies to target butyrophilin 3A1, and then to interrogate its efficacy in vivo, we utilized immunodeficient mice. So butyrophilin 3A1 is not expressed, or its homolog is not, its ortholog is not known in mouse. So we had to use completely humanized systems. So in order to interrogate its biology in vivo, we utilized immunodeficient mice that we implanted with human ovarian tumors that express an antigen of interest. We infused these mice then with antigen-specific T cells to target these tumors. And you can see when we co-treated these mice with adoptive transfer of antigen-specific T cells, along with our antibody that we had identified that could target butyrophilin 3A1, we had a suppressed malignant progression. Obviously, the tumor still progressed, but this did have an impact in suppression progression of these tumors. Importantly, not only it suppressed progression, but it outperformed nivolumab. So it outperformed the clinically approved antibody to target PD-L1. So clearly, butyrophilins have a role in regulating anti-tumor immunity. I believe it's not only specific for ovarian cancers, we've also seen expression in breast cancers and other malignancies as well. So, a lot of the failed responses to checkpoint immunotherapies may be due to we just don't understand enough about the unique environments of each cancer that may be regulating anti-tumor immunity. But we're overall excited about butyrophilins as a potential novel checkpoint molecule, and we are actively pursuing additional investigations of other butyrophilin molecules. So we're currently interrogating the butyrophilin-like family of molecules, which are structurally similar to butyrophilin 3A1. And just briefly, what we've seen so far is that butyrophilin-like 8 is also immunosuppressive through this in vitro system that we've generated in the lab, and which is a relevant target as it is highly expressed in ovarian cancer. So we're excited about butyrophilins overall as novel immune regulators of anti-tumor immunity in cancer. So, carrying on with our interest in understanding the regulation of anti-tumor immunity in the context of cancer, I became interested a few years ago in what other how else T cells may be suppressed. So we know that checkpoints are critical, but the local tumor microenvironment has also been shown to dictate and suppress T cell activity due to environmental factors. Importantly, endoplasmic reticulum stress has been recently shown to regulate anti-tumor T cell activity. So just quickly, ER stress is defined by the accumulation of misfolded proteins in the ER that are sensed by IRE1, PERK, or ATF6 that can induce translocation of transcription factors to drive programs to respond to this stress response, right? So in cancer cells, this has been shown to be tumor promoting. But a few years ago, I was involved in a study where we found that ER stress driven by IRE1 XBP1 activity could actually suppress T cell activity in ovarian tumor beds. And this stress is likely driven by oxidative stress, hypoxia, nutrient deprivation. where it was shown that tumor-derived factors could impair the uptake of glucose from the environment. That led to defective glycosylation, drove ER stress, and impaired the ability of the T cell to uptake glutamine, and that drove metabolic changes that were detrimental to T cell activity. So it was kind of fortuitous that I was working on that project at the time, because I was, asking questions, what else is regulating T cell activity? And I was doing so at the time by interrogating TCGA data sets, basically to identify potential novel factors that have an outcome, a differential outcome for women with ovarian cancer. And one interesting hit that I found was in this molecule known as UHRF1/BP1. And this came to my attention because there was a polymorphism, as you can see here. Let me switch to there we go. There's a polymorphism here in the red, right? So that those patients that possess this polymorphism had a better overall outcome compared to ovarian cancer patients. So these are high-grade serous, mostly ovarian cancer patients that do not express this polymorphism. So this is a germline mutation. It's predicted to be loss of function. The really interesting thing about this polymorphism, though, is that it's associated with the development of systemic lupus erythematosus. So up until a couple of months ago, the only thing in the literature about this molecule was it was a risk loci for SLE development. So clearly that hinted that this molecule may have a role in regulating immune responses, and potentially, in the case that in the data we see here, maybe regulating anti-tumor immunity to promote hyperactive T cell responses or any kind of leukocyte response that may be protective for the host. As I mentioned, this is predicted to be loss of function because this particular polymorphism is found in a highly conserved region within the gene, shared amongst a variety of species. So we began to, how do we begin to interrogate this molecule? So, nothing is known about it. Nothing is still known about it. So we're working on this on a lot of fronts. But does UHRF1/BP1, irrespective of the polymorphism, but the biology of the molecule itself, how does that potentially regulate T cell activity and T cell activity that may protect the host against cancer? So we created a conditional knockout model in case, if people here aren't familiar. This is a classical Cre-Lox system where we can knock out a gene specifically in a tissue of interest through a promoter-driven Cre recombinase, which can basically excise a gene by recognizing these LOXP sites. It basically cuts, the functional part of the gene out, so that we have the conditional knockout mouse. And in this case, we use a CD4 promoter, which basically knocks out UHRF1/BP1 out of all peripheral T cells, CD4 and CD8. When we utilize these mice and we challenge them with a murine model of high-grade serous ovarian cancer, this is actually a very aggressive model of high-grade serous ovarian cancer. We observed a significant survival advantage for those mice, suggesting that the ablation, that this molecule may be regulating anti-tumor immunity. And if we quantify T cells in these tumors, these tumors possess more T cells. That's generally associated with better outcomes. More importantly, though, if we go to our tumor bank and we create cohorts of patients that express ancestral wild type UHRF1/BP1 or those that express the polymorphism, we identified, as we saw in our mouse model, greater infiltration of T cells into these tumors as well. So this is a point that we're performing additional investigations to understand the quality of these T cells. So there's a greater frequency, but we're currently interrogating the phenotype of these T cells. We're undergoing single cell RNA-seq to understand how differential populations may be functional. And we're also characterizing potential antigen specificity, as well as TCR clonality to get a better handle on how UHR from BP1 may be driving the survival advantage through modulating the T cell repertoire. So, those initial studies, we performed RNA sequencing, this is total RNA sequencing, and, based upon what we found in the mouse model, it's not surprising that we found that there are signatures associated with basically T cell proliferation, T cell activity, as well as responses to cellular stress, which was interesting at that time, as well as elevated glycolysis. So if we take those T cells from those tumor beds with mice that had the conditional knockout mice or wild type mice, if we isolate T cells from those tumors and re-stimulate them with antigens, again, those T cells respond more effectively to antigen re-stimulation through the production of interferon gamma and granzyme B. So these are essentially the knockouts. We have a nice polyeffector phenotype, as well as a greater ability for those T cells to proliferate, suggesting that we have that UHR from BP1 deficient T cells had this effector phenotype, which is more greatly associated with outcomes in, it's associated with better outcomes for patients with ovarian cancer. And interestingly, if we go to the seahorse and we see a greater utilization of extracellular acidification, as well as the release of lactate, suggesting that these T cells better utilize glycolysis than their wild type counterparts. But what is the mechanism, right? So we have a nice phenotype. How is this functioning? And as I mentioned, not much is known about UHR from BP1. If we align domains of this molecule with other with molecules of somewhat known function, so UHR from BP1 shares a N-terminal domain, this chorion N domain that's associated with the VPS 13 family, as well as ATG2. And what's known about this domain is associated with lipid transport and trafficking. So there's a potential that the function of UHR from BP1 facilitates trafficking of lipids from one membrane to another, and we're interrogating this through a variety of mechanisms. So what we decided to do at that point was then to really begin to understand what potentially may be occurring here. Because we went back to our RNA sequencing data, and T cells that had intact UHR from BP1, so this is compared to the knockout T cells, had a greater utilization of pathways associated with redox homeostasis, glutathione peroxidase, peroxisome activity, as well as oxidoreductase activity. So we wanted to then interrogate what were the binding partners of this molecule. So we utilized Jurkat cells, which are a immortalized T cell line, basically pulled out UHR from BP1, and then we performed mass spec. And interestingly, we found an association of this molecule with the mitochondria, with vacuolar proteins and peroxisomes. So the association that I found that potentially may be occurring here is that mitochondria, upon cellular stress, have been shown to communicate with peroxisomes through the formation of utilization of vacuolar complexes through what are known as microvascular bodies. And we confirmed the association of UHR from BP1 with the mitochondria here. So given the our pathway analysis showing that there were a potential dysregulation of redox homeostasis, as well as pathways involved with peroxisomal activity, we began to think that perhaps the activity of the enhanced activity of UHR from BP1 deficient T cells may be driven by redox, an imbalance in redox homeostasis that may reside in a dysregulation of peroxisomal activity. And I'm trying to get my knowledge of peroxisomal biology increased here, but what's known about peroxisomes is they are a critical site for detoxification of hydrogen peroxides through the activity of catalase. So this is a mechanism that we are potentially we are currently exploring. So how may this be occurring in the context of cellular stress? And I'll try to pick up the pace here a little bit, but if this is in fact if the absence of UHR from BP1 is actually causing a redox imbalance, it makes sense that potentially this would be a stress response molecule. So I'll skip the details of the graph here, but what we've seen so far is when we expose T cells to FCCP, which induces oxidative stress through mitochondrial uncoupling, we see an increase in transcription of UHR from BP1, suggesting that cellular stress through the integrated stress response system may drive UHR from BP1 transcription. And if this is the case, then if we block this with an integrated stress response inhibitor, we should see a decrease in this transcription, which we do see here. This all occurs the integrated stress response system all occurs through the activity of this transcription factor known as ATF4. So we interrogated the promoter of UHR from BP1, and we found potential ATF4 binding sites that may be driving this transcriptional response. And in fact, when we perform CHIP PCR, we do see ATF4 localized to the promoter of UHR from BP1, specifically upon the induction of mitochondrial stress, suggesting it is a mitochondrial stress response element that's transcription responsive to this stress that may be facilitating redox homeostasis. And when we stress T cells with a relevant tumor microenvironmental stressor such as hypoxia, we again see increased transcription of UHR from BP1. And this occurs in association with an elevated level of reactive oxygen species within T cells. So UHR from BP1, it appears to be a stress response element that may facilitate homeostasis between the mitochondria and peroxisomes that drive peroxisomal detoxification of ROS. So ROS are important because they're known to be potential drivers of secondary TCR signaling. So moderate levels of ROS can facilitate TCR activation through a variety of mechanisms. So to interrogate, does our phenotype that we see associated with our UHR from BP1 knockout mice, so this elevated effector phenotype, so if we isolate T cells again from these tumor beds and we re-stimulate them with antigen, we see that again our UHR from BP1 knockout T cells have greater interferon gamma production, suggesting they are more effective, right? But when we introduce a ROS scavenger in acetylcysteine to the system, this increased effector activity is blunted. So this does suggest that this redox system that UHR from BP1 appears to dictate when it's dysregulated may be facilitating this elevated effector function as we scavenge ROS, this effector activity is lost. So we came up with this model, it's a little bit in the weeds, but that tumor microenvironmental stress, hypoxia, or perhaps other stressors can drive mitochondrial stress. So these are known mitochondrial stress response elements through ATF4 that drive UHR from BP1 transcription to regulate redox homeostasis, and which again regulates T cell activity in tumor beds. However, if this molecule is not functional, this imbalance in ROS activity can drive elevated effector function and perhaps glycolytic reprogramming. So what are the therapeutic implications? So we're thinking about how we can utilize this system to, essentially treat patients. So what we've done so far is basically adoptively transfer primed T cells into mice and into tumor-bearing mice. And what we've seen is that that the transfer of UHR from BP1 ablated T cells protects these mice to a greater extent. And this is in the context of elevated PD-1. So these mice do express these knockout T cells do express elevated PD-1. So this suggests that we get the survival benefit in the knockout condition, But if we combine this with PD-1 blockade, perhaps this will drive a greater survival benefit. But really, what we want to do is interrogate this in the context of human immunology. So we're going to utilize cancer specimens from ovarian cancer patients and basically implant them into humanized immunodeficient mice, as well as to sort and expand their reactive T cells. We're going to create cohorts that express the polymorphic variant of this molecule or the ancestral variant, and then we'll monitor outcomes in the context of, in addition to checkpoint blockade as well, to understand the potential clinical implications. So I went over a little bit, I think, so sorry about that. I just want to thank my group really quickly, as well as our collaborators and our funding sources.